Hi everyone, I'm really excited to be talking about GRouNdGAN, which has this idea of letting GRNs causally drive the simulation of single-cell RNA-seq data. My name is Yazdan, and I'm a student at McGill. And let's start today's talk with a quick background on gene regulatory networks, or GRNs. As you know, gene expression is controlled by interactions of genes and their various regulatory elements, such as transcription factors. And we can think of these interactions as a graph and also model GRNs as such. And very often, we are simplifying GRNs into bipartite graphs. And this model has gotten us pretty far. And because of that, there's been quite a lot of interest in reconstructing these GRNs from either bulk or single-cell data, especially from single-cell data in the past decade. And with that, a need to systematically benchmark these GRN inference methods. Now, how do we do that? We need two components. First is a single-cell RNA-seq data set, and second is a corresponding ground truth GRN. And here's where we face a challenge, because how do we come up with this ground truth GRN? Well, we could try to curate interaction from literature, so either manually or through text mining. That's very prone to error. For example, you might end up with GRN edges that are missing. Or we could rely on structured biological databases of interactions or on ChIP-seq data sets. But the common problem with all these ways of aggregating interactions into a GRN is that the final GRN might not even correspond to our biological system under study. Because as you know, gene regulation is very context dependent and also cell type specific. So the only way to really ensure that our GRN corresponds to our system is by performing perturbation experiments on that same system. And those are very long and costly. So the next logical step would be to use synthetic data for benchmarking, but we also face challenges here because most simulators for single cell don't incorporate this GRN and instead focus on being general purpose simulators with a focus on generating realistic single cells. So they cannot be used for such benchmarking. Now, there are very few simulators such as BoolODE that actually do incorporate the GRN. BoolODE was used by a study called Beeline to benchmark a number of GRN inference methods. And Beeline also benchmarked those same methods using experimental data, but they found that some of the best performing methods on simulated data were the worst performing ones on the experimental data benchmarks. So that's a gap that we want to close. Furthermore, current simulators that are GRN guided require on the user to perform some kind of post hoc distribution matching once they generate data that corresponds to a certain GRN. Now, that process on its own is very subjective, but there is also some unknown effect that it introduces when it comes to the causal effect of TFs and genes that was previously imposed. And finally, they all have some simplifying assumptions when it comes to how the combination of TFs work. For example, in the case of two TFs co-regulating a gene, they make very simplifying assumptions. So with GRouNdGAN, we wanted to have a causal implicit generative model that does not suffer from all these issues that I just talked about. The way GRouNdGAN works is through a two-step training procedure. So in the first step, we pre-train a Wasserstein GAN with gradient penalty given a reference data set to simply generate realistic looking data. And over here, the critic is trying to score how realistic its inputs are, while the generator is trying to generate cells that the critic thinks are real. So once we train this first Wasserstein GAN, we isolate its generator, we rename it to a causal controller, which now only generates transcription factors. And these TFs are used as input to a second GAN's generator, which is composed of many units called target generators. So each target generator generates a specific gene. So if we look at gene 3 over here, it is being regulated by TFM and TF1. And the gene 3 target generator over here receives as input TFM and TF1. And that is how, in the second step, the GRN is imposed onto the model in a way where every single gene is being generated under the control of its regulators. And we train this second GAN, once again, as a Wasserstein GAN, with the addition of two auxiliary networks, which help us further impose the GRN and causality onto the model. But how does GRouNdGAN perform? Well, we tested it on various single-cell data sets. Here I'm showing the results on only two of them. So we have Tessany plots over here with real cells being in blue, generated ones in red, and we see that they kind of overlap in this 2D space. And even the smaller cell types in the PBMC data set, which are in those little islands, although GRouNdGAN didn't receive any cell type info, it doesn't suffer from this mode collapse problem and still generates those. Aside from this, we have a number of quantitative metrics to see how realistic the generated cells are, and we compare it to the state-of-the-art simulators, including scGAN and scDesign. And we are already familiar with some of these metrics, but I wanted to introduce the RF-AURC score, which is essentially when we train a random forest to distinguish between real and generated cells, and we want to fool it so it performs close to random. And GRouNdGAN is successful at fooling that RF. However, our only goal wasn't to generate realistic cells. We also wanted to causally impose the GRN. And how do we test for causality? Well, one way of testing for causality is through performing interventional experiments. So in our case, we have a GRN, and for example, we can intervene on one of its TFs and see how that affects its target gene expression values, and if that change is significant or not. And the way that works in the model is by setting 0 one of the TFs that are outputted by the causal controller. So here I'm setting TF1 to 0, for example, and keeping all the parameters constant, doing a forward pass. And when we do this one TF at a time, of course, genes that are not being regulated by that TF, their expression remains unchanged. But genes that are regulated by that TF, their expression changes, and we perform statistical testing to quantify that change. So here we have this histogram of adjusted p-value for that experiment, which shows pretty significant p-values. So now we have a model that generates realistic data that imposes a GRN causally, but we now want to see if a GRN reconstruction method is capable of inferring that GRN that is imposed into the data, and what happens to the edges that were present in the data, but we either failed to impose them or simply did not want to impose them. So to answer those questions, we here design some kind of controlled experiments where we first start by finding the top 10 regulating TFs for each gene from experimental data. For that, we use GRN-Boost2. And we then create 2 separate GRNs by subsampling that scaffold GRN. So the positive control GRN, the one we imposed onto the model, is comprised of that each gene and its TFs ranked 1, 3, 5, 7, and 9. And the negative control GRN has interactions between that gene and its TFs ranked 2, 4, 6, 8, and 10. So these 2 GRNs are comparable in importance, have similar density, as it's shown by this green dotted line, which is the PR curve of GRN-Boost2 applied to real data, evaluated on positive control on the left and negative control on the right. And we see that when we now infer GRN on GRouNdGAN generated data, which imposes the positive control, those edges are emphasized, and GRN-Boost2 and other inference methods are able to assign them higher scores. However, edges that were not imposed through the model, the ones in the negative control, shown in the blue line in the right figure, those are now assigned lower importance than they originally had in the reference data set, which showcases that GRouNdGAN is not only emphasizing edges that were imposed through it, but also messing up and with the interactions that are implicit to the data, but we did not want to impose. Now, where can we obtain this scaffold GRN, which we then impose onto the model? We already talked about how we can get it from literature or from biological databases. Our approach here is to learning that scaffold GRN from experimental data and using a subset, for example, of identified edges as positive control and the other subset as negative control. Now, a very important note here is that our goal is not to learn the actual causal GRN of the reference data set. Our goal is to simply learn a set of interactions that are consistent with the data, although they might correspond to spurious correlations. How? Because once we take those edges, create a GRN and impose it onto the model, those edges become the causal edges of the synthetic data. to showcase how that would work, for example, here we constructed that scaffold GRN using PP-core, which is a purely correlation-based method for GRN inference. And when we now infer GRN on the synthetic data using GRN Boost 2, once again, we're able to recover a certain GRN. So the edges that were purely correlation edges in the reference data are now turned into causal edges in the synthetic data. And we use the same controlled setup of positive-negative control to benchmark numerous inference methods that were also present in the Beeline study. And our results pretty much exactly match the results from Beeline when it comes to how these GRN inference methods ranked. But our results were consistent with their results on experimental data and not with their results on synthetic data. So we can say that using GRouNdGAN, we have bridged this existing gap, which is between synthetic data benchmarks and real data benchmarks. Now, some of the inference methods that we talked about use this concept of trajectories and pseudotime. And oftentimes in single cell, we want to preserve trajectories and study different cell states. So to test if GRouNdGAN is also preserving those trajectories, we train our model on a bone marrow data set, which has stem cells differentiating towards different lineages, and using PAGA construct these trajectory graphs. And we see that for different lineages, the marker activation patterns are very similar between real and GRouNdGAN simulated data. And to extend on this notion of trajectories and pseudotime, we also perform pseudotime inference using diffusion pseudotime on both real and real reference and simulated data. And over here, we see the activation of some lineage markers and maturity markers over the pseudotemporal x-axis. And once again, highly concordant between real and simulated data. So to conclude, here I presented GRouNdGAN, which is a simulator for single cell, which generates realistic data and does not need any user interventions. It can do this process automatically. It preserves gene identities and pseudotime ordering of cells in a way where genes are causally expressed under the control of regulating TFs. And we saw how it can be used to perform these kind of in silico perturbation experiments. If you're more interested in that, check out our paper. We do some actual more interesting experiments on that front. And very importantly, bridges the gap between synthetic and real data benchmarks. So if you're interested, this is our preprint on bioRxiv. And there's also a code base and tutorial available on GitHub, which shows you how you can train and also use GRouNdGAN. And with that, I'd like to thank all my lab members who are in this picture. And if you have any questions, I'll be very happy to answer them. Thank you.
Great talk and great work. As we know, the performance of GRN inference methods depends on the nature of correlations among the TFs themselves, because in real data, they're very correlated. That's part of the reason why GRN inference is hard. So when you generate the TF expression profile in that first phase, have you seen whether the correlations among the generated TF expression are similar to what exists in realistic data?
Yeah, so you're referring to this first step of training the model, which is generating TF expressions.
That's right.
Now, in this first step, we are simply trying to generate realistic data, because right now we're not imposing any GRN onto the model at this step, right? So the only focus is to generate realistic looking data and essentially mimicking our reference data set. So if the TFs are correlated in the reference data set, they will be correlated in this generated data set. It would be interesting to actually quantify that change. We haven't done it for specific TFs and see if they're correlated, but the different metrics that we have, here I'm showing some of the metrics for just all TFs and genes combined. But I personally have also done it on the subset of TFs instead of focusing on TFs and genes. And it shows a high degree of similarity with the reference data set. So I believe that.
Great answer. Thank you.
Yeah, thank you.
Hello, I'm Jessica Rexak. I'm faculty here at UCLA. We've been using footprinting from corresponding single-cell ATAC data as a way of benchmarking GRNs, and I was curious whether you've tested that against the GRouNdGAN and seen whether it's corresponding or.
So if I understand correctly, using footprinting from ATAC-seq to, no, we haven't
validate.
used other modalities yet. We're trying to integrate new modalities and generate those as well. But our goal here was to have some kind of tool that is very systematic, which is also mathematically guided, that actually has a causal graph, that has a guarantee that a causal graph is imposed onto the model. Now, it would be interesting to definitely look at ATAC-seq and see how, if it's concordant or not, but we still haven't done that.
And then the last question I had has to do with TF interactions, right? I think it's, in my mind, it's still relatively open how the interactions of different TFs might alter their binding. And typically, we don't think about it that much, but there's a whole class of small MAF family genes that interact with FOS and a bunch of, and depending on those interactions and their co-expression, it completely changes their binding. And those binding motifs are actually not well annotated in the databases. So anyway, any thoughts on that? Because you have an interesting tool here that could maybe be applied to think about that.
Well, that would require us to essentially fundamentally change the GRN that we're trying to learn, because here we're trying to learn a bipartite GRN.
It would require a line between two
Exactly.
TFs.
So it would need TFs to be related. However, in GRouNdGAN, these TFs are not independent of each other, because in the first step of training, when we're simply trying to, generate realistic-looking cells, we generate one cell at a time in a way where each TF, which is being generated by this first generator, is not independent of the rest. Independence between genes only comes in the model in the second training step, where we now have to impose a GRN and have these target generators which do not have any connections between them. However, speaking of in theory, since we're generating all TFs at once in this first step and then isolating them, the interactions between the TFs, that line in this graph, that should still be captured by the model. I don't see why it wouldn't be.
But does that change the ability to then transfer that onto another system? Or does it, because you're always going to go back to the beginning and reassign those TFs as a group for the next analysis, it's fine?
So for this second training step,
Mm-hmm.
right?
Oh, sorry.
Yeah, so over here, the
Separately.
TFs are not independent, but in the case that are, for example, co-regulating a certain gene, I believe that will be as close as we can get to real biology, since the TFs are not independent of each other. However, if two genes are being generated with a different set of TFs and regulators, then they will have independence, some kind of independence in terms of expression.
Thank you. I just got enthralled. Forgive me. Thank you.
No problem. Okay, in the interest of time, we should move to the next